Hey future doctors, welcome to Pocket Mbbs, where medicine fits in your pockets. Now let's study today's topic, that is staining methods. In microbiology, we will study the different stains. Now the first is Gram stain, which was developed by Hans Christian Gram in 1894. It is most widely used test for bacteriology, and its procedure includes fixation, means the smear is made on the slide from bacterial culture or a specimen in air is dried and then heat fixed. Now the steps of Gram staining are: step 1, primary staining, in which the crystal violet is added for 1 minute and then rinsed with water. In step 2, a mordant is added, that is gram iodine, for 1 minute and then rinsed with water. In step 3, decolorization, acetone is added for 2 to 3 seconds, which is 95% ethyl alcohol based, and then we wash it. Now in the step 4, which is the counter stain, water is rinsed immediately and dilute carbonyl fuchsin or safranin is poured for 30 seconds and then it is also rinsed with water. Now there are the two results that we get, and in case of carbonyl fuchsin or safranin we can also use neutral red. Now the observations are, that is, first, if it is a gram positive bacteria, then it will resist decolorization and retain the primary stain, that is violet color, and if it is gram negative, then it gets decolorized and takes the counter stain and appear pink. Now the principle of gram staining. That first theory is pH theory, in which the cytoplasm of gram negative bacteria is more acidic, hence can retain basic dye, that is crystal violet, and iodine serves as a mordant, that is, it combines with the primary stain to form a di-iodine complex which get retained inside the cell. So in this way the primary stain is retained in the gram positive bacteria. Now the most famous theory, that is the cell wall theory, in which gram positive bacteria has thick peptidoglycan layer which is 50 to 100 layers thick and itself is not stained, and instead it acts as a permeability barrier preventing loss of crystal violet, and large di-iodine complex not able to penetrate the thin layer. Whereas in the gram negative bacteria there is a thin peptidoglycan layer and no tight linkage of the presence of lipopolysaccharide layer due to presence of lipopolysaccharide layer in the cell wall, which easily disrupts by decolorizer, forming large pores that allows the di-iodine complex to escape from the cytoplasm. So in this way the gram positive bacteria retain the primary stain and gram negative bacteria takes the counter stain. Now the third theory is magnesium ribonucleate theory, in which it is present of cell wall, that is gram positive and gram negative, and help to retain the primary dye. Means magnesium ribonucleate is present in their cell wall, which will help gram positive to retain the color and gram negative to lose the primary stain color and take the counter stain. So these are the three principles of gram staining. Now the uses of gram stain is to differentiate gram positive and gram negative bacteria. It gives idea for further biochemical test for further identification of bacteria. It is also used to start the empirical treatment with broad spectrum antibiotic before culture report available. It is also useful for certain fungi like Candida and Cryptococcus. It helps in screening quality of sputum specimen processing for culture. Modifications of gram staining are: it was modified by Jensen modification, useful in meningococci and gonococci. Brown and Benning modification, used for actinomycetes. Then gram positive bacteria examples are Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumoniae, Bacillus anthracis, Clostridium tetani, Clostridium botulinum. Gram negative bacteria example are Escherichia coli, Klebsiella pneumoniae, Salmonella typhi, Proteus mirabilis, Vibrio cholerae, Neisseria gonorrhoeus. Now we will see the acid fast stain, which was discovered by Paul Ehrlich which is further modified by Ziehl and Neelsen. It identifies acid-fast bacilli. Ziehl-Neelsen technique has hot method, in which the step 1 is primary stain, in which a strong carbonyl fusion is added for 5 minutes and intermediate heating is done by flaming underneath the slide for better penetration of the carbonyl fusion into the cells of the bacteria, and then it is rinsed with water. Now in the step 2, decolorization, in which 25% of sulfuric acid is used for 2 to 4 minutes. It is placed in the slide and then proper decolorization occurs and rinsed with water. Now step 3 is counter staining, in which 0.1% methylene blue is used for 30 seconds and then rinsed with water. Now the interpretation is that Mycobacterium tuberculosis appear long, slender, straight, slightly curved red colonies, and it is the acid-fast bacilli and non-acid-fast bacilli organisms present in the background, and it takes the counter stain color and appear blue. Now the acid-fast bacilli examples are Mycobacterium tuberculosis, Mycobacterium leprae, and Nocardia, and the sulfuric acid percentage used for these bacilli are 25%, 5%, and 1%. Now the modifications of acid-fast staining. Earlier hot method is used and now various cold methods are used. First is Quignon's method, in which Quignon's carbonyl fusion is over the smear for 5 minutes. Now decolorize with acid alcohol for 3 minutes, then counter stain with methylene blue for 4 minutes. In Quignon's method, no heat is done. Now in Gabbitt's method, it is also a two-step method in which the acid basic add basic fission, that is phenol stain, for 10 minutes, then counter stain with Gabbitt's methylene blue for 2 minutes. Now the principle of Ziehl staining is acid-fast bacilli rich in mycolic acid, which makes them resistant to decolorization by acid alcohol after staining with strong carbonyl fusion. That's why acid-fast bacilli do not take the counter stain, and heating is done so that carbonyl fusion penetrates the cell wall and enters the cell wall of the bacteria. Now we will study a third type of stain, that is Albert stain. It is used to demonstrate the metachromatic granules of Corynebacterium diphtheria, and its procedure is after heat fixation of the smear, it is poured with Albert stain 1 for 5 minutes. Then Albert stain 2, which is iodine solution, is added for 1 minute, then rinsed. Then wash the slide and bottle dry and examine under the microscope. Then we will see the Corynebacterium diphtheria in the microsite. Now the composition of Albert stain are: Albert stain 1 comprises the toluidine blue, malachite green, glacial acetic acid, alcohol 95% ethanol, and distilled water. Albert stain 2 contain iodine in potassium iodide solution. Now the interpretation is Corynebacterium diphtheria appear green colored bacilli arranged in Chinese letter or cuneiform pattern with bluish black metachromatic granule at polar ends. Now here we can see Gram staining, in which we see Gram positive and Gram negative bacilli. In acid-fast staining, we see acid-fast bacilli, and in Albert staining, we see Corynebacterium diphtheria with Chinese letter appearance and microchromatic granules. Now, thank you. That's all for today's videos. Follow for more such videos and keep growing my channel.